Thank you, Roman. It's really a pleasure to be here. Thanks for organizing this workshop, Roman, Christos, Manuela, and Peter. I really enjoyed it. And today I will tell you about the work that has been going on in our group, where we mostly study entanglement, how topology affects equilibrium, kinetic, mechanical properties of polymers in general, biopolymers. And I will tell you about some studies that we did over the years regarding the entanglement of DNA, how it can be probed, why it is interesting, and also point at some recent works where also the presence of electroosmotic flow, therefore ions, interaction with charges and in the flow, and DNA plays a very important role in determining the mechanical response that you can probe in single molecule experiments. So essentially, this is the outline of the talk. I will slightly invert the order of these two and first tell you how you can probe the entanglement, the knotted state of DNA, and then mention which are the applications of that, and therefore why it can be important to have such tools. And I would like to introduce you about the, let's say, the go-to technique to detect the knotted state of DNA by starting with a seminal paper. It's a paper produced by the group of Cozzarelli back in '93. They considered some linear DNA. It is a viral genome. It comes from a specific bacteriophage, P4. And the DNA of this bacteriophage is 10 kilobases long. It is a double-stranded DNA, but is terminated by short single-stranded termini, which are complementary. And therefore, when these termini have the chance to meet, they anneal and they circularize the DNA, not in a covalent way. It is nicked, therefore you cannot store torsion there, but you trap the molecule in a circular state. So when you put the molecule in solution and it circularizes, if the molecule before circularization happened to be entangled in a physical knot state, in an open chain, after circularization, then the knot is trapped as a proper topological state, so what also mathematicians would call a knot. And you can detect how many and which type of knots you have by doing gel electrophoresis experiments. You produce a very dilute gel, you put an electric field across it, of course DNA is charged, and DNA migrates with a velocity that depends on the topological state of DNA. So this is, let's say, a non-conventional use of electrophoresis, which the most common application is to resolve molecules that have different migration velocities because they have different lengths. Here they are monodispersed in length. This is genetic genomic DNA, but they travel with different velocities because they are tied in different knot types, and therefore the different type of topological complexity makes the molecules engage the obstacle of the gel in different ways. And by measuring the intensity of the bands, you can detect what type of knots that you have. And people are quite sure about those because they checked the knot types using AFM microscopy after coating DNA to make it thick enough to be resolved. And they know which bands correspond to which knot types. Okay, so first, maybe I thought that besides using this as an introduction, I might also show a couple of figures from the paper of Cozzarelli, which I think might be of interest to the community here, because one thing that they did back in the day was to check what is the impact of the strength of the ionic solution, monovalent salt, the sodium chloride, on the knotting probability of the DNA in equilibrium and on the abundance of the various knot types. So for example, in this plot here, taken from the paper, here you have an increasing concentration of let's say positive counter ions. And here you see that they measured the amount of molecules that in equilibrium after circularization are in the trefoil knot state, the simplest type of knots, and for these DNA lengths, the most abundant. And you see that it increases steadily. And to have an interpretation, a microscopic or let's say mechanistic interpretation of that, they resorted to a very simple but very useful coarse-grained model. They treated DNA as a chain of cylinders, fully flexible chain of cylinders. So the cylinders they took to be equal to the Kuhn length, twice the Kuhn length, and then they also used shorter lengths of the cylinder with appropriate bending rigidity to reproduce the overall persistence length. And essentially they had only a limited combination of parameters. Only one 8-dimensional parameter is important. what is the effective diameter of the cylinders that you consider with respect to the length of the cylinder, and then you can scale it up, not to your Kuhn length if you wish. And therefore, as a function of this parameter, within the model, you can compute what is the probability of having knots. So if you wish, more prosaically, if you have a rope of a given length, as you make the rope thicker, it becomes more difficult to tie knots with it. No? So this is essentially what you see here. So it's a matter of relative thickness to length. And by using this as a guideline, they were able to check when the knotting probability that you measure matches data seen experimentally. You scale it up by the Kuhn length of the DNA, and what you get from the knotting probability is a measure of the effective thickness of DNA. So for the various salt concentration, what is the effective thickness of DNA that accounts for the observed equilibrium knotting probability? And you can see that you recover in a neat way, but through this more complex topological route, essentially the Debye-Huckel screening length, which here happens to be the potent length that control the features. So I introduce you this phenomenology and also the technique that has been used mostly to detect knots in DNA. And the reason why that technique is important, not only, let's say, for conceptual reasons, but also for practical reasons, is that it gives you a way to probe indirectly what is the organization of DNA in complex situations, such as inside the capsid of viruses. So this is the P4 bacteriophage, the same one from which the P4 DNA was coming from. It has a capsid of about 50 nanometers in diameter. It has a long neck, a very narrow diameter. The genomic DNA is all stored in there. One of the sticky ends is anchored here at the foot of the virus. And the question, ready to be injected inside the bacteria that is, when it is infected. So people were able to use the gel electrophoretic technique to probe what is the entangled state of DNA. You cannot really see what happens there. You can use cryo-EM, but you have to do averaging, and therefore what you get is an indication of the average order that you have there, not the topological disorder, which is what people were interested. And so they were able essentially to take advantage of the circularization of the genomic DNA of the virus to trap the knotted state in the capsid, do the gel electrophoresis experiments, and they established that within the capsid, the DNA is knotted, physical knots in open chains, in more than 95% of the viral particles. So you have these, and then you have also specific knot types. So if you have a model for how DNA should be organized inside a capsid, then you should be able to match these knotting probabilities, which we and other groups have worked on. So this raises all sorts of interesting questions. For instance, how come that you can afford to have DNA that is highly knotted inside the capsid and is still capable of coming out from this narrow exit channel when it has to deliver it to a bacterium? And then other questions. How can you push the limit of this gel electrophoretic technique beyond the length of 10 kilobases that you see here, which is a practical limit of application for these techniques? So these are more fundamental questions. This is a more technical one. I would like to touch upon, I give you the answer about this conundrum first, because I think it is a nice phenomenology. So imagine, the intuition of us and everybody is that since you have a highly knotted DNA inside, it has to come out from a channel that is wide enough to allow the passage only of a single double strand. If you have knots, you would expect them to be stuck, no? By the time that they reach the entrance of the pore and not be able to translocate. But actually, our intuition was wrong. The same phenomenology has implications also for the sequencing that people are nowadays doing, Oxford Nanopore and other companies in sequencing DNA, where they fish filaments of DNA in solution. They can be very long. They can be therefore knotted spontaneously, and you translocate them through a pore that is narrow enough to have only one single-stranded filament going through. So you do unzipping at the same time as you translocate. And therefore, if your molecule has knots, so what happens when you try to translocate these molecules to a very narrow pore? It's a similar situation where, in this case, the pooling is done by the application of an electric field across the pore rather than the pressure, the entropic pressure that you have from the capsid going to the outside. So let me give you the outcome without the theory of our simulations. This is a fully flexible polymer of 15,000 beads. It is knotted. It contains a figure of 8 knots. We didn't put it there, so we just sampled the equilibrium ensemble of configurations of fully flexible polymers. At this length, the knotting probability is a few percent, so it's not very hard to fish them. If you make twice as long, you saturate exponentially fast to 1. And then if you try to translocate the molecules, so here is the slab. This is the pore. You put a field here that drags the molecule. So the molecule is pulled by a force that is applied only here. This is what you see. So the molecules are knotted. I will play the movie again because there are several things that go on. But the main thing is that what you see is that the molecule goes through the pore with no hindrance whatsoever. The knot is there. When we saw the data, we thought that the knot had disappeared. Maybe there's been bond crossings. We pulled too strong. No, the knot is there. So there are all sorts of interesting things that go on that are fairly well understood from unknotted polymers. First of all, the force that is applied only here propagates and invades the chain, propagates through a fractal embedded in three-dimensional space. Therefore, as time progresses, translocation is slower and slower and slower. But apparently, the presence of a knot does not make a difference. The knot initially was somewhere here. Therefore, if we look at the close-up, the knot was up here. And if you look what happens at this close-up, now you see the knot is here. It has been dragged down to the pore. It stays there, tightened, and the chain is able to reptate along its knotted contour and essentially going through without apparent hindrance. This is because thermal fluctuations are these forces, which are of the order that we expect to be at play, for instance, in the DNA inside the viral capsids. The thermal fluctuations are enough to let the knot breathe a little bit. So by applying a force, you can take advantage of that and gain bead by bead by bead, and it goes through. But this also gives you the intuition, a posteriori, that if you apply a much stronger force, now this is almost 10 times larger than before, now you overcome the thermal fluctuations. Now, essentially, you develop so much friction there that now the molecule is not able to translocate and reptate along its contour anymore, and you recover the macroscopic intuition that this thing eventually should not be able to stop. So this is for a fully flexible chain, but the results translate all the same if you use a more detailed model for DNA with intrinsic stiffness. So we use the oxDNA model. Essentially, you see the same phenomenology. As a function of time, the number of base pairs that translocate initially goes up, as you would expect in a chain without any topological hindrance. And it goes up. As you make the force stronger, you go through faster. But eventually, if you keep increasing the force, you create this friction there, and the molecule is not able to translocate effectively anymore, and then you stall it. So here, essentially, this addresses this question here. There was the other question that I mentioned is, can you apply better techniques than gel electrophoresis to address longer lengths that are not addressable with this method? And pore translocation has been used to this purpose too. The next slides are taken from the beautiful experiments carried out almost 10 years ago by Kiesdecker and co-workers. They used solid-state nanopores, fairly wide, so the diameter was about 20 nanometers, so much wider than the width of the DNA, and wide enough to allow knots to go through, unlike the Nanopore that I considered before. They put an electric field, and they cannot, of course, observe directly what is going on, but they can measure the flow of the current, so the ionic current that goes through the pore. You have a baseline level where there is nothing inside the pore. when a linear DNA filament goes through, you see the current going down by 1 notch because you have the occupation of the double-stranded DNA, excluded volume here inside the pore. If you have a signal where you see initially that you go down by 2 notches and then 1, and then you go back to 0, it means that transiently you had 2 double strands that go into 1, and so the interpretation is obvious. You have a backfolded end, that entered first. It can be at the end and so on. And then you have signals like this one that require to make sense of. You go down by 1 notch, meaning a linear DNA is entered, and then suddenly you see a spike that goes down by 2 notches. So all of a sudden you have 3 DNA strands inside the pore, and then you go back to 1. And the interpretation of that is that you have caught a knotted DNA, because if you have, let's say, the simplest type of knots, if you take a cross-section of the knot, that corresponds to 3 strands. And therefore, this is a method that is scalable to lengths of 100 kilobases and even longer in their study, that allows you to detect the presence of knots in DNA, and therefore can be used for various types of probes and to answer various types of questions. What is the knot improbability? You just do the counting of what you see. You can also address where the knot is positioned, presumably, because you just look when this event occurs, at least that is the expectation. And by looking at the duration of the event, you might be able to say how long the knot is. If knot is very tight or longer, you would expect signals that are longer or smaller. At least that is the intuition. Then the phenomenology complicates things or makes them more interesting according to perspectives. What they saw, what they reported, is that if they looked at when the passages of knot were happening, they saw that they were mostly happening towards the end of the translocation process. So there was a bias. You would expect them to be uniform, maybe, but they saw a bias towards that. Not only that, the distribution of durations was very broad, possibly indicating that you had knots of very different sizes, possibly. Modeling allows you to make contact with these experiments and provide an intuition that otherwise it's not obvious to have by just thinking about the problem. So we used an oxDNA model. So this is a filament that gets translocated through the pore, but if you look closely, this really has still simplified, but the double helical nature of DNA, because we didn't know whether in the presence of the knots that gets translocated, maybe you had the local melting of the DNA and so on. So we wanted to have a better model than just a chain of bits. So we used the oxDNA. This is an example of a simulation. The red region is where a knot is. You can track the beginning and end of the knot along the contour here. You see that now the knot has tightened as it translocates. It makes a few diving attempts until it successfully goes inside the pore, and then translocation progresses. Okay, so here is how translocation progresses, fraction of translocated chain. Here is the knotted region. So what is important about this? What does this video add to the expectation that we saw before? Well, the translocation process alters significantly the properties of the knot compared to the initial equilibrium state. By the time it translocates, the knot has tightened, and so we can explain this bias towards late translocation stages of DNA, which you can reproduce in simulation, due to the fact that as you translocate the chain, the knot tightens and typically tightens upstream because it has to be reached by the propagating tension. Okay, so that explains the bias. What you measure is not the properties of the knot undisturbed. Okay, and then how about this broad distribution of translocation times? Well, it turns out that, as I pointed out in the simulation that you saw before, translocation does not proceed with uniform velocity, but slows down more and more and more because you're propagating tension in a chain that is embedded as a fractal in 3D. And this slowing down that you can see from these curves here reverberates in having a broad distribution that has not very much to do with the fact with the knot being large or small. knots are always tight, but they translocate with different velocities because translocation typically happens quite late and therefore with a low velocity that gives you these long translocation time that are not indicative of tight knots. And on top of that, there are various modes for which knots can translocate. They can be tight, as you saw in the simulation, but they can also present themselves in about 50% of the cases in this other geometry here. It also gives the same signal because essentially you have all the crossings, what I call essential crossings, still localized in space. As you pull them through, you still get a blip in the signal of the current or a drop in the speed of the current. Although here you would say that the knot is delocalized over this entire region here. But then the essential crossings are still localized, and therefore this one here, only this bit contributes to the drop of the current. Past the essential crossings, when you are still inside the knot, it would look like an unknotted circular DNA. So, in the last 5 minutes, I want to tell you more about an effect that we recently established in collaboration with the group of Ulrich Kaiser, where electroosmotic flow, as I anticipated, plays a very big role. The setup is similar to the one used by Decker, but the pore size is now intermediate between the very narrow pores and the very wide pores. The pore size is about 10 nanometers. And the nanopores, in this case, I'm not sure if it makes a difference, but there are detectable differences compared to cylindrical solid-state nanopores. The nanopore is obtained from melting a glass pipette. So the nanopore has a conical shape, which can play a role probably in the features that you observe here. You put a potential difference across, which drags the DNA through, and the setup is the same as what you measure is the current, as in the experiments that you saw before. One feature that is immediate, already before discussing the experiment, is you see that in this case, the signal that corresponds to this detection of this tangle here schematized by a knot, that corresponds to a very broad signal, very different from the spike signal that you saw for the tight knots. Okay? So that's already telling that you have something complicated going on. But then, more blatantly, if you use that setup to measure the knotting probability of DNA of various lengths, as you see here, that is way above what you expect in equilibrium, way above. The longer the chain, the more you see. You see twice as many knots that you expect to have in equilibrium. And it is very unlikely that over a timescale of the few milliseconds of the experiment, you are introducing knots, because that timescale is much smaller than the relaxation timescale of DNA, let's say of 50 kilobases. So it's very unlikely that you are introducing knots. And what you are observing the signal much in abundance. Not only that, but for a given knot chain length, 50 kilobases, that is lambda DNA, as you increase the voltage, as you pull it stronger and stronger, faster and faster, you see more and more knots. I should say more and more tangles, something that gives the same signature of knots, these drops. For more than a year, we tried various models, say, okay, maybe the persistence length in this ionic condition is different, but it can be different only up to a point. There was no way to reconcile these experiments with the models until we came across at a conference at a work that had been carried out in the group of Aximentiev, and they did a fully atomistic study of double-stranded DNA in a narrow pore in the presence of an electric field. This is with explicit water, explicit ion, and counter ions. And what they observed, and then it has been confirmed experimentally, is that as the DNA is pulled through the pore, it also starts to spin because it is a chiral molecule embedded in a flow of ions and also counter ions. It is like, I don't know what is the English name, but this toy that kids, that spins, no? When kids start to run, you have a chiral object in a flow. Of course, it has a preferred directionality. This holds for DNA, but holds also more microscopically. This is from a recent paper of Decker. With origami, he constructed a microscopic chiral, mesoscopic microscope, okay, whatever, you see here, a few nanometers, chiral object, it also starts to spin, it is a rotor. So this gave us the idea of checking whether this can be an important role. You can imagine what can happen. So we didn't have any explicit counter ions, no explicit water, but we did put the torque effect besides the translocation effect parameterized after Aximentjev's runs. And this is what we saw. So this is the initial equilibrated configuration of DNA. It is a chain that can sustain torsion. And now there is a torque that is applied, and you see that you have the formation of the plectonemes that Christos was mentioning yesterday, except that now they are plectonemes in an open chain. So being open, it's not subject to the Calagariano-Fuller theorem of conservation. It can dissipate torsion through the ends. Okay? So let me play it again. You can see what is the relevant phenomenology. Plectonemes form, they don't necessarily make it through the pore, so there is a certain transmission coefficient. Some of them can dissipate along the way, but this is a new phenomenology, no, that appears. And therefore, indeed, what you can check is that through simulations, and I think this is the last slide, through simulations, you can check that indeed plectonemes appear. They appear with the correct probability that you need to account for the missing knots, knot counts. But more importantly, they come with a specific signature that tells them apart from knots. They are being very long. They are associated with long drops of long duration. And this is also what you see experimentally. Experimentally, you see two types of signals, short and long. And so our proposal is that the long ones, that is not so difficult to get, are plectonemes, and the short are truly the knot ones, and then you can reconcile the features. Okay, so probably you can read my conclusion faster than I can, and thank you for your attention.